Thank you very much, and I think it meets the EU expectation to move from the basic science to industry, so we saw here many examples. So thank you very much, Professor Gazit. Our next speaker is Professor Yosi Diamand Shacham, the Bernard Schwartz Chair of the Nanoscale Information Technology, Department of Physics, Electronics at the Faculty of Engineering. But I must add something, that the link between the two first speakers is one of the pioneering project of the EU, the pioneering network of excellence, Nano to Life, that you saw so many pictures at Professor Gazit lecture. And the Nano to Life was actually a network of excellence of 20 research centers in Europe 10 years ago, and 23 industries. And the core of this network of excellence was the joint research collaboration that was led by Tel Aviv, and I had the privilege to do it as the coordinator, and actually created a platform that yielded later more than 100 funded projects, and it's going on and on up till today. And you know, both the first lecturers, both Professor Gazit and Professor Shacham, were having projects that were a kind of an outcome of this network of excellence. And by involving 20 industries, it brings over the utilization of this research. So Professor Shacham, please.
Thank you, Mira. I need to find my presentation, but you may need to help me because I don't know how to operate Windows.
Okay, so should I help you?
I think this is the one. Good. And I need to push something. Is it correct?
Yes, see.
I'm a Mac person.
I don't believe it.
I'm a Macintosh person. I don't know
So
how to
what?
operate.
If you're a Macintosh I mean, you're doing science.
This is what I'm doing. Thank you, Mira. And thank you for the very kind introduction. And I really have to admit that when I hear the name Nano to Life, I have very good feelings. It was a turning point in our university that we were introduced to this group of wonderful people in Europe. And I have to really thank Mr. Patrick Boisson from France and Françoise Charbit and many other good friends, which became very good friends. And my career started in a completely different place. I was working for many years in VLSI technology. I'm from engineering. My knowledge in biology and life science was very limited. But as I became the director of the Nanoscience and Technology Institute in our university, and I was very lucky to meet these wonderful people from Europe, and we worked very hard, but nevertheless, I said we were lucky. But for luck, you need to work. And we won the grant. And we really put a lot of efforts led by Mira, that was our right hand and left hand and the brain and the action. And she did wonderful things. And we were introduced to life science. And I will show that I even changed my career. I started to work with biologists. And in the last 15 years, I'm learning biology. And I decided, since I worked for many years on interfacing electronic circuits between themselves, my research is in interfacing machines and interfacing microprocessors and memories. I decided why not to interface it with biology. Signal is a signal. It can come from a computer. It can come from the brain. And Mira, I'm not sure if you mentioned, one of the projects that we had was RenaChip project that we specifically interface the brain. So I'm in the next 18 minutes I have left, I will give you kind of a short overview. What does it mean, Nano-Bio interfacing? And again, my perspective is from engineering. My group, I think, is one of the few groups that students develop their process, work with UDI labs and Professor Amichai Freeman and other people, take protein and put them on chips, but they also design the circuits. We design amplifiers and analog digital system, and also build systems. Today with the buzzword of Internet of Things, you try to put everything on the network, and try to connect them together. So my group is in engineering, but I have students in my group sitting together, biology, chemist, physics, and recently material science, our new department in engineering. And I was very happy that Udi Gazit was introduced as his research is in materials, which I think he's also becoming an engineer a little bit. Interfacing biology. So interfacing biology, the key is sensing. What sensing is you take something, some physical quantity, you convert it to a signal, can be optical, can be electrical, can be mechanical, but you take something from life, and this is the key. You can take physical component, but you can take biology, and this is my, and excuse me the biologist, this is my impression. You have, biology is like a lot of artifacts, some are blue, some are green, some are, and today I want to detect the blue square. So I build a sensor to take the blue square and find it. If it's a glucose for diabetes, I want to detect glucose. And this is the key. You build sensors, you interface them, and once we try to abstract it, to make this, a signal is a signal. And sometimes I have to tell you that sometimes we look for signals and we develop our own algorithms, and after 10 years we find out that other people are actually working on it in other disciplines. So the key is for me as the advisor of the students is to know many things to make sure we don't miss anything from other disciplines. And again, if you want to put it today, you want to put it on the cloud architecture. Today you want to link them together with all these wonderful words. And this is kind of, this scheme show from left to right, a lot of sensor connected to antennas, connected to routers, connected to the cloud. At the end of the day, you are sitting with your computer or smartphone or something, and you want this information, and you want it yesterday, and you don't know how you live without it. So, talking about recognition elements, you have antibodies, enzyme, DNA, whole cell tissues. This is kind of a hierarchical concept. And what we do in my group, for example, if you want to interface body or interface components, you need to make electrodes. Electrodes are these components which take the signal and convert it from the body, from biology, but you need to build an interface, like brain research, peripheral nerve signal pickups. Other interesting issue is instead of moving the electrodes inside the body, is move the cells on the chip. So taking cells, cancer cells, toxic, sensing toxic materials in water, bring the object onto the sensor. And the last thing I'm not going to talk today because we are very limited in time, but the sensor can also do some actuation. We can do something, not just sense, but we can do something. This is for a different talk. We have some projects, European projects on this also, but today we focus on sensors, like the RENA-Chip project with the collaboration of Professor Mati Mintz, Hagit Messer-Yaron, and Mira. It was ISF funded, and also FP7, the RENA-Chip consortium. This is just a short list of the members, University of Newcastle. I'm not going to read the whole details, but every project was an opportunity to find wonderful people in various disciplines and to build a system. And the system, this is kind of our, if you want to ask what is RENA-Chip, is you put an electrode into the brain, into the cerebellum, in the what we call the small brain in Hebrew, if you translate it from Hebrew. And you excuse me if I'm not using the right terms, but if you put an electrode into the cerebellum, you can actually pick up signals. After training, this was implanted in rats, transfer it into the computer, and store this information in the computer. And if something goes wrong, you can program the brain to do this function again. And in my group, we designed the electrodes, we designed the interface. This is a picture of the real electrode. It's made, to produce this, we use our expertise in microfabrication of electrodes, which are used for microprocessors, but in this case, we use the same technology to make electrodes to pick up. The actual signal is in the tip. This is a large magnification of the tip, where you can actually put the electrodes next to a single neuron and pick up the signal from neuron. And this was a wonderful experience, and these are the signals. The signals, look, don't ask me where is the signal, but Professor Hagit Messer-Yaron and with the help of Mira, they managed to take this, something which looks like noise, and with very clever signal processing, they actually showed us after a while that here in the second, you can see something. While in the top, you don't see anything. And I'm not going through this whole process, but at the end of the day, they managed to pick up. We took the signal, transferred it to a computer, and they managed to get the signal. So this RENA chip project ended, let me go back, successfully with a lot of, the key technology was achieved, but we also developed some side effects, for example, with Dr. Aryeh Taub, which is my student and Mati Min's student. He's now a researcher in the Weizmann Institute. We developed some specific coating preventing the scarring on the electrodes. For example, if you put an electrode into the brain, after a while, you have some scar tissue. You have some the body wants to reject the electrode, so we developed a technology which can be useful for other technologies. And this is something which you may see. Sometimes you start a project, and you end up with some technology which is helping you on your project, but can be useful to many other projects, which is now a topic for another project. We also developed sensor and the SmartHand project, and this is a group of people. The coordinator was in the Lund, Jan Sibelius and Thomas Laurel, and from Italy and from other countries, and we developed this what we call special electrodes. It was one of the first flexible devices that were made in Israel. Now there's a lot of activity in what we call flexible technologies. What you see to your left are flexible electrodes which are implanted on peripheral nerves. And this is how it looks. We introduced some organic materials, conducting polymers. But without going to details, we managed to produce electrodes that can be wrapped around the nerve and pick up signal from nerves to pick up signal, what we call the phantom signals, which were used to realize to realize the information being transferred to an artificial hand. I'm going from project to project to show you our role. We are developing the chips, the technology of the chips, and we work with biologists. Another interesting project was, and this is a collection of projects that is now, we are now celebrating like 15 years to this collaboration with Professor Belkin from the Hebrew University, Amichai Freeman from our university, and Judith Rishbond. We were partners of many projects, including not only the EU, we also were funded by DARPA and local projects. And in the last 15 years, what we do, we are trying to pick up signals directly from cells. And those are optical signals, electronic signals, mechanic signals. And what we try to ask the cell, how do you feel today? I mean, if there's a microbe, do you feel bad? Do you feel, what's going on? How was your day today? So what we focused in our group is in this specific process when some analytes affect some promoter, some part of the genetic material in the brain, generating some, through the, being translated to some protein, and we detect this protein. And our expertise in the last 15 years is detect these proteins. They have wonderful names like alkaline phosphatase, all these biological names. But the key, you want to detect this specific molecule when it happens, and when it happens, you know something going on. So one of the projects is the whole-celled sensor for rapid cancer sensing and screening. This is the list of the participants of this integrated project. I intentionally put it in small font so you cannot see it because it's a really long list. But we got a wonderful collaboration. The result with some local researcher, we actually developed what we call like the Tau system that if you look what we do, if you put some cancer cells or normal cells, we can differentiate between cancerous tissues and normal tissues by measuring some electrochemical processes. Specifically, for example, for colon cancer, we detect alkaline phosphatase, which is a biomarker. In melanoma cancer, we detect an enzyme called tyrosinase, which is a marker. And as an engineer, I'm not going through these specific chemical processes today, but the idea was, let's develop something that everybody can use in 5 minutes. You take a tissue. Today, you have to send it to biopsy. It's a process that takes a few weeks. It costs money. And in developed countries, sometimes it's not a big problem because people, have this infrastructure, but underdeveloped countries or third world countries, there is no center. You want to know it, and we developed systems that are kind of a wholesale Nano-Bio sensors. It's a biochip. You take a biopsy or take a small piece of the skin, put it on the system, take a coffee. After 2 minutes, you know the result if this tissue is cancerous or not. And this is just to show this how the signal looks like. We measure the current after some specific reactions. And we actually did some experiments in hospital. This is the results you get from the hospital after 4 weeks. This is our result. And we are practically 100% like what is called the golden standard. This is the sensor for tyrosinase for melanoma. This is the bottom line is the signal for a normal cell. The top line is for a sick tissue. This technology now is working in the lab. We are trying to transfer it to experiments in hospital. The last part of the talk today is the talk about the water toxicity sensor, which is instead of looking at human tissues, we look at microbes. And we have many projects detecting, the left is the list of the things that we do. We do detect genotoxicity. We also like to detect acute toxicity. And if you want to do it, you have to put molecules or cells or organism or sometimes a population. Some people say that ER, emergency room, is a wonderful sensor for toxicity, because if you see many people coming to the hospital with the same toxical effect, you know something wrong. But we don't like to use humans as sensors. It's not recommended. So we try to put it on a chip. This is another project, the Toxi chip, and this is the list of participants. And it's actually, we have few projects, one for optical sensors. This is another project with the Federal Ministry of Education in Germany, on electrical sensors with this, if you can read German, it's Bundesstaat für Gewasserkunde in Koblenz, and excuse me from a German accent. We are developing sensors. The goal is a simple device for toxicity sensors, like the same, all these things. We took some pictures of things you buy in the supermarket. We want to have a device that you're in the field, you want to drink the water. Can I drink the water or not? You don't have time to send it out for analysis. So the solution is integrated wholesale biosensors, genetically engineered microbes, broadband sensing, solid state platform, all these electronics. And what we actually do, the left side, if the toxin material hits the microbe, there's chain of events in the microbes. If there is a signal, it means don't drink the water. If there is no signal, you can drink the water. So far, I talked about mostly electrochemical effects. A parallel channel is to use electrical effects, and this is kind of an observation. There are many papers on biosensor using functional electrodes. However, there are very few of them which are used. So we want to develop some to understand why also detection fail, like impressive detection in the lab, and they don't work. And this is a big problem in my field. And one of the keys is to overcome the barrier of transferring from university to the industry to be practical, to be useful. This is kind of a concept of what we call the sensors. Sensor have some sensing element. And if everything goes well, this is what you detect. But sometimes you have some defects which you also sense, and this is an interruption. So what do we study? The loss of selectivity, instability, all these engineering terms that we actually build sensors, and the long-term effect, noise, signal loss, biofouling, scar tissue formation, electrical problems, and leakage. And we also sometimes, previously I showed you that only electrodes, but you can also, this is a transistor, we can also integrate transistors as part of the sensor. This is our new research. We are, again, looking for now for funding for this kind of research. And if this is the last slide, if you think about it, everybody talks about the Internet of Things. So the next step is to integrate all these sensors. And if you look, this is kind of, it's not a reality, it's kind of a joke, but in the left side today, you buy the sensors, and this is what I call wearable 1.0. My watch is the wearable. in the end of the day, we want to integrate it into the structure. So you cannot see it, but they will be part of you. And this is the next step in the sensors that we do, what we call printed sensor, functional sensors, printed flexible sensors. Thank you.